Hello everyone, thank you for attending the oral defense. My name is Xiaoyu Zhan from Beijing University of Technology. It's so glad to report my work at the IC2020 conference. And today my topic is a computer-aided diagnostic system to detect polyps in computed tomographic colonography images. We will introduce the whole thesis in 5 parts. As the first, background and motivation, give a brief. Second part is going to show image processing conceptual model. In the third part, the specific polyp detection model based on hybrid features will be further introduced. And the fourth part is experiment. Finally, we get the conclusion. With the improvement of living standards, more and more people suffer from colorectal cancer. According to the data released by the National Cancer Center, the number of cancer patients in 2015 was about 3.929 million, and the number of intestinal cancer cases was 388,000 in China. There are no obvious symptoms in the early stage of intestinal cancer, and it is difficult to identify intestinal polyps due to the complex internal environment of human intestine. The picture shows the structure of human intestine. Doctors are likely to miss or misstart the polyps due to a large number of folds and undulations in the large intestine structure. Compared with colonoscopy, CT colonography has the advantage of less invasion, and CTC has the same superior effect as colonoscopy. Moreover, in the medical imaging processing, computer-aided system is a universal system to assist the doctors to find lesions that may be missed in CT images when diagnosing diseases. From the above, it is meaningful to propose a CAD model for detecting polyps in CTC images. By reviewing the studies, we found that there are some gaps in the research of CTC image polyp recognition CAD system, especially in the part of CTC imaging polyp classification. Most of the existing research focuses on the traditional machine learning methods, which usually come from the relatively global region, and the specificity of its features has not been studied for specific diseases. The study of CTC polyps with deep neural networking focus on the local features and the neglected overall characteristics such as tumor volume and size are also very important in clinical practice, which may lead to confusion in clinical decision making. The field of image classification based on machine learning method is becoming mature. The computer distinguishes different kinds of objects through different image informations of different feature depth. The image processing conceptual model is shown in the picture. The original image data set is pre-processed to enhance the related information, After image pre-processing, we get the optimized image dataset. After feature extraction and feature selection, we get the birth feature collection. Finally, we get the result by classification algorithm. And then, based on the conceptual model, we propose polyp detection model based on hybrid features. In our method framework, we extract the HOG feature set from the ROI region after pre-processing the image dataset. After that, we use the relief algorithm to assign weights to the feature set and then select the high-weight global feature set. The C for optimized classifier SVM and RF are used to classify and compare the result of combined feature set. At the same time, we divide a home image into patches. In the patches, we use the PCNN model we defined to select the optimal contribution patches set in each image and then obtain the local feature sets extracted by them. When getting global features, we extract the HOG features first. Its basic idea uses the gradient information to reflect the age information of image target and turns the appearance and shape of image into characterization data by the size of the local gradient. The relief algorithm selects a sample random. The feature weight is obtained by respectively calculating the distance between the sample and the nearest class neighbor of the same category and the nearest neighbor sample of the different category. Finally, it selects the weight greater than the first formula calculates the weight of feature A. The second formula calculates the distance between sample on feature A. In our experimental method, we choose the C algorithm to optimize the penalty parameter C in the classifier SVM and the number of trees in RF to obtain the optimal value. Chaotic vector is a key step in the chaotic evolution algorithm. At the first, the chaotic vector direction factor and the chaotic parameters are obtained randomly, and then each individual is evaluated by fitness function. Then mutation and crossover are used to generate chaotic vectors, and each new chaotic vector is also evaluated by fitness function. Meanwhile, in order to judge whether the chaotic vector is suitable to join the generation, the chaotic vector is compared with the target vector by greedy selection. We do not cross the target vector and the mutation vector when optimizing. Thus, the formula for generating the chaotic vector is as the formula shows. The PCNN model is used to extract the local features. L2 is the loss function of PCNN. Afterwards, we insert the patches dataset into our CNN model to obtain the optimal patches feature set. Our PCNN model consists of a 5-layer convolutional layer for feature extraction and 2 fully connected layers for classifying future maps. Some parameters in the PCNN model have already been set as a table. Others will be compared in the experiment. In the experimental setting, we get 100 polyp samples and 140 non-polyp samples. By data cleaning, at last we have 168 images in the training set and 72 in the test set. Accuracy is used to evaluate the experimental results. In extracting HOG features, we set the number of cells in each block to 1 and the pixel of each cell to 32, which can extract more details of the features. When using the relief algorithm to choose features, select n features with weights greater than 0 and use SVM and RF classifiers to examine the classification results for the first key features, and the value of key is between 1 and n. PCNN model will compare the results of different parameters. The classification results of different feature numbers shows that RF classifier is superior to SVM classifier in the feature set. At the same time, RF classifier is the best when the number of features is 35. In the PCNN model, we get the parameters with both the experimental result. Finally, we obtain the local feature set with 256 features, and the birth result is 0.83783. The results with different methods was shown in the table. Our hybrid method was the best among the above classification methods with an accuracy of 0.91318. This proves the superiority of the CAD method for CTC images constructed in our paper. In the research, we established a CAD model for automatic detection whether CTC images contain polyps for detection and diagnosis. We first extract the HOG feature set, then use the relief algorithm to select the global feature set, then obtain the local feature set by our PCNN model with the selected patches set, and find the RF classifier. Classification result is better than the SVM classifier. By comparing the classification result with only global features or local features, our hybrid method achieves better classification results. Thank you very much for the privilege of presenting this paper.